So, in today's lecture, we will continue our discussion on gel based proteomics, certain advantages and challenges of performing DIGE experiments, how to analyze DIGE data by using few very specialized software, and how to interpret that data, how to obtain some meaningful biological information from those analyses. So, today I have invited a guest, Dr. Srinivas from G Healthcare, who is going to talk to us about DIGE technology and give us a demonstration on software to perform DIGE gel analysis. This is my great pleasure to introduce Dr. Srinivas Rao from G Healthcare. He is an application specialist in the research product division of G Healthcare. Dr. Srinivas, thank you for coming for discussion about 2D and DIGE technologies. So, what are the major advantages and disadvantages you see by using 2D approach?
It's very user friendly as well as very low instrumentation cost. There, the complete proteomic, the differentially expressed proteins we can see exactly during this complete 2D electrophoresis technique, as well as the novel proteins we can able to isolate from from this technique. This we can identify very easily the softwares as well as very user friendly. This is what we can do. There are some disadvantages. The reproducibility will be not there in most of the times. This is a major disadvantage of this 2D electrophoresis. And again, softwares, they are very user friendly. Still, there is lots to need to be developed so that we can use very effectively.
So, as you rightly mentioned, so two-dimensional electrophoresis is still a very powerful tool to resolve thousands of proteins on the gel.
Yes.
One can also analyze isoforms and post-translational modifications, including phosphorylation, and overall the process is very user-friendly.
Yes.
So, still it is one of the very powerful technologies currently available for proteome analysis.
Yes.
Now, I also agree with your comment that there is some reproducibility issues because users have to run the gels from the control and treatment separately, and then there will be a lot of manual artifacts regardless of how meticulous they are doing the experiment. So, in that regard, what is your suggestion, like how user can overcome the limitations of traditional two-dimensional electrophoresis technology?
One should have to prepare very good sample preparation so that the reproducibility thing will overcome. The second point is, these days the new generation 2D electrophoresis system, that is the DIGE technology, is available, where you can use 3 samples in a single strip and we can co-migrate them in a single strip, and we finally there will be an internal control as well as control and treated. These 3 you can run in a single strip, so that these kind of reproducibility issues we can overcome. As well as there is a scanner, very powerful scanner is available in this technology, that is the Typhoon Trio, where you can have this is based on the laser based technology, this completely, so that you will have very efficient gels you will have in your hand. This is
so as you mentioned that DIGE technology is one of the very powerful solution to overcome several challenges which people face in running traditional 2D gels. And so in that regard, I think if you can elaborate a little bit about DIGE technology, so that will be useful to the students.
Yes, sir. I have a small video regarding the DIGE technology. I'll show you that video so that everyone can able to easily understand what exactly DIGE technology. Yes, as we were talking about the 3 samples we can label with Psi2, Psi3, Psi5. Now you have 2 different samples from different source. Now you are taking these 3 samples into 3 different Eppendorf tubes and you are labeling with Psi3 dye as well as again another with Psi5. Then you are taking those 2 samples, pooling them and labeling with Psi2. then you're mixing those three into a single tube, then you're running in a single strip. There you will be analyzed in first dimension the IEF, whereas in first dimension after analyzing IEF you will be placing into second dimension where you are completely analyzed the second dimension. Now this gel can be scanned in the Typhoon Trioplus, where you will have 3 images from a single gel. Then this can be analyzed with the help of DIGE software, then where the complete analysis can be done with the help of DIGE. Then in DIA there will be a co-detection. The co-detection nothing but there is a standard gel which is labeled with Site2. From the same area we are expanding remaining 2 images also, so that the artifact can be minimized. Again, this is fully automated analysis. There will be very less manual interference would be there in this analysis. And after finishing of DIA, we can go to BVA. In BVA, we can compare all DIAs together. Like as you can see in this video, this complete first of all it matches between the standard gel, and from standard gel to again the corresponding DIA. It matches each and individual spot, and it will give you the final data. Then these BVAs can be analyzed in the further in EDA, that is extended data analysis. Here you will have lots of stringent statistical data, that's a differential expression analysis, pattern analysis, discriminant analysis, and PCA analysis.
So Dr. Srinivas, it was very useful to get the glimpse of the overall process in DIGE technology. Would you like to elaborate or demonstrate some more detail about steps involved in doing the labeling of this technology?
Yes, there is another video where you can see now. This is the second video which you can see more elaboratively. This is particularly a protocol which is developed for membrane protein analysis. Now you can see this video.
In the cell surface labeling protocol, seen on top, you label the cells while they are still intact. During the labeling process, the dye will only have access to the cell surface proteins. After the labeling step, the cells are lysed. To verify cell surface specific labeling, the labeled sample was fractionated into membrane and cytosolic proteins. A non-fractionated sample was prepared in parallel for comparison. This fractionation analysis is not necessary, but was done here just to show that the cell surface protocol is specific for cell surface proteins. We also performed the standard 2D-DIGE labeling protocol, seen below. The cells are lysed before labeling, and in this way all cellular proteins are accessible for labeling. After the labeling step, the samples are subjected to 2D electrophoresis. Adherent cells are detached using a non-enzymatic procedure to avoid digestion of the cell surface proteins targeted in this protocol. We use the rubber policeman, but using enzyme-free cell dissociation media is also an option. Count and divide the cell suspensions into aliquots of 5 to 10 million cells per tube. The cells are then pelleted and washed in HBSS pH 7.4 to remove traces of cell culture media. Contamination from serum proteins and fluorescent components can interfere with labeling and detection. Cells growing in suspension are directly pelleted and washed before the labeling step. After the wash, the cell pellet is resuspended in 200 microliters ice-cold labeling buffer containing HBSS pH 8.5 and 1 molar urea for optimal labeling conditions of cell surface proteins. Always check pH before labeling. We used 600 picomoles Sci-Dye for 10 million CHO cells. The optimal ratio of Sci-Dye to cell number will vary depending on the cell type, since we do not know the exact protein concentrations on the cell surface. How to determine the optimal conditions for Sci-Dye labeling of proteins is described in the 2D Electrophoresis Principles and Methods Handbook. The cells are incubated with 2D-DIGE floor minimal dyes for 20 minutes on ice in the dark. After the labeling reaction, the unreacted dye is quenched by adding 20 microliters of 10 millimolar lysine. The labeled cells are now washed twice in cold HBSS pH 7.4 buffer to remove the excess Sci-Dye. There will therefore be no free dye left for unwanted intracellular labeling of proteins in the next step, which is cell lysis. The proteins on the cell surface are now labeled and the cells are washed and ready to be lysed. The pellet from the last washing step is resuspended in 150 microliter cold lysis buffer containing 7 molar urea, 2 molar thiourea, 4% chaps, 30 millimolar tris, 5 millimolar magnesium acetate pH 8.5 and left on ice for at least 1 hour with occasional vortexing. The samples are now ready for 2D gel separation. The first step in 2D electrophoresis is to prepare IPG strips for rehydration. Prepare the streak rehydration solution by adding IPG buffer corresponding to the pH interval of the strips used and add the solution in the lanes of the rehydration tray. Remove the protective film of the IPG strip and place the strips carefully with the dried gel facing down in the rehydration tray containing the rehydration solution. Close the lid of the IPG box and rehydrate the strips overnight. In the first dimension, isoelectric focusing, the proteins are separated according to their pI. This is performed using the IPG4-3. The rehydrated strips is placed in the manifold and the electrode is mounted on top. 50 micrograms from each sample were applied using sample application cups. We have either directly applied non-fractionated samples without prior fractionation or fractionated the samples into membrane and cytosolic fractions before they were applied. The lid is closed to protect the fluorescent samples from light. The instrument was programmed according to recommendation and run overnight. Large 12% Lemley gels were cast using a Dalt 12 gel caster. Displacing solution was added to avoid polymerized acrylamide gels in the tubings. The gels were allowed to polymerize overnight at room temperature prior to use. After the isoelectric focusing, the strips are removed and equilibrated in SDS-containing buffer in 2 steps using DTT to reduce the disulfide bonds of cysteine residues, followed by alkylation with iodacetamide to avoid modification by acrylamide. The IPG strips are dipped in running buffer and carefully placed on top of the large 2D gels. Avoid trapping air between the strips and the gel. Seal by adding melted 2% agarose solution with bromophenol blue on top. The gels are now ready for second dimension SDS page separation. In the second dimension SDS page, the proteins will be separated according to molecular weight, and this is performed with the ethanol 6 system. Fill the electrophoresis chamber with anodic running buffer, insert the gels and fill the top compartment with cathodic running buffer. Program the power supply according to recommendations and run the second dimension protected from light for approximately 4 to 5 hours or until the dye front reaches the bottom of the gel. After the second dimension electrophoresis, the gel cassettes are placed by using the grippers in the Typhoon fluorescent imager. 2 gels and 3 channels can be scanned simultaneously. The result from this 2D gel shows high resolution of membrane proteins in the sample, even if there are some known restrictions for hydrophobic proteins to be detected in a 2D gel. The results show many new cell surface label spots, shown here in red, that are not detected using the standard labeling protocol, shown here in green. These results show that the cell surface labeling protocol is highly specific for labeling cell surface proteins. Since cell surface proteins are exclusively labeled, they are more easily visualized and attenuation by abundant cytosolic proteins is avoided. The fluorescent image of gels with non-fractionated, membrane fraction or cytosolic fraction is shown on top. Below is an image of the same gel post-stained with silver. The results show no fluorescent labeling of cytosolic proteins, but the silver staining shows that there are proteins in the gels. The results also show similar spot map patterns from non-fractionated and membrane fractions, demonstrating that there is no need for fractionation prior to 2D electrophoresis, which makes this protocol both simple and convenient. Cy2, Cy3 and Cy5 show similar labeling patterns and are all compatible with the cell surface protocol. DIGE experiment was performed using all 3 2D-DIGE floor minimal dyes for studying differential expression of cell surface proteins in CHO cells after serum starvation for different lengths of time. Cy2 cell surface samples from all samples in the experiment were pooled and used as an internal standard. The differential changes of several cell surface proteins could be followed during the starvation period. Over 80 novel membrane proteins were detected using the cell surface protocol that were not detected with the standard DIGE protocol. For finding the identity of the proteins in the preparative gel, it was necessary to spike with a cell surface sample to facilitate matching back to the spots on the analytical dataset.
So, Dr. Srinivas, it was very useful to see all the steps which are involved in doing this DIGE technology. There is that discussion going on in the proteomics field that due to advancement in mass spectrometry and microarrays, how do you foresee the future of two-dimensional electrophoresis and DIGE technology? Do you think it's going to be simultaneously working along with the mass spec and microarrays, or do you think it's going to be behind because now more advanced approaches have emerged? Yes. So what is your suggestion on that?
Yes, the MS and microarrays develop differently. They have their own advantages as well as disadvantages. Like disadvantages like very high instrumentation cost and high protein requirement, as well as technical variance would be there. That is why still 2D-DIGE will be the core technology to analyze proteomics. One can easily analyze.
So you are still very convinced that by using 2D or DIGE approach, that will still remain one of the core proteomic technology for proteomic analysis.
Yes, definitely.
And it will be used simultaneously along with mass spectrometry and microarrays.
Yes.
So at the end, I would like to request you to give a brief overview of the DCider software which is being used for analysis of DIGE gel.
Sure.
DCider is the software to analyze DIGE gels, where we can analyze DIGE gels in different stages. In first stage, we have to upload our gels into our software. This is basically works on the Oracle database, where we are incorporating our gels into database. There you can add these gels into software by this way, and you can select what are the gels you want to upload, and once you have uploaded, then you can able to edit these gels here itself, like you want to any crop or edit anything, any sort of things you can do here. It will allow you to edit these images. So now you can see this is the overlay of 3 images. Now you can crop all these images, like whatever the portion you want to take. If you have to remove any area which is not your interest of, then you can directly crop those images by this way. Once you directly editing, you can save it, yes, save it and close it. This is what you can edit these images. Then you have to create a new project. Suppose your project name would be according to your project name. This is demo. Okay, now we have the project name of demo. Now you can able to import these 2 gel images into a new project where we created. Just click on import. Now these gels will be imported into this particular project. Now here you can see all 6 images. Though these are the basically 2 gels, inside within these 2 gels we have Cy2, Cy3, Cy5 and total 6 gel images. Okay, this is the image editor and image loader.